I think we'll go ahead and get started. So it's my great pleasure to introduce our speaker today, Professor Taija Mäkinen. Professor Mäkinen is an internationally recognized leader in vascular and lymphatic biology. She currently serves as director of the Wihuri Research Institute and professor of translational medicine at the University of Helsinki. Her research has significantly advanced our understanding of how blood and lymphatic vessels develop, function, and contribute to human disease. After earning her PhD at the University of Helsinki, Dr. Mäkinen completed postdoctoral training at the Max Planck Institute. She then established her independent research program at the London Research Institute of Cancer Research, and then later joined Uppsala University. Throughout her career, she has made pioneering contributions to the study of vascular and lymphatic morphogenesis, and lymphatic vessel development, and the mechanisms underlying lymphatic disorders. Today, Dr. Mäkinen will share her latest insights into this exciting area of research, and the title of her presentation is Cellular Dialogues in PIK3CA-related Vascular Malformations. Please join me in welcoming Professor Taija Mäkinen.
Okay, thank you so much, Mike. It's really a great pleasure to be here, and I will today, as Mike said, I will discuss our work on PIK3CA driven vascular malformations, focusing, of course, on lymphatic malformations, but if there is time in the end, I will also discuss briefly venous malformations. And so what I would like to really focus on is how these, as we know, the disease-causing mutations in endothelial cells not only affect the behavior of those cells, but also how those mutant endothelial cells then communicate with the microenvironment to drive disease progression. And how, by better understanding of these mechanisms, we may be able to develop new therapeutic opportunities. And with a very short introduction, which is surely you are all familiar with, but I will just give some general background that will be, some of it will be important later in my presentation. So as you know, the cardiovascular system is traditionally viewed as a kind of a systemic distribution network formed of kind of conduits of for blood, nutrients, and cells. But beyond this classical role, we now know that vessels, and in particular the microvasculature, are increasingly recognized as active tissue communication interfaces. So they continuously interact with their surrounding microenvironment. And importantly, the vasculature, and especially the microvasculature again, shows this remarkable tissue-specific specialization, both at the level of different types of vessels. So in the blood vasculature, of course, arteries and veins, and in the lymphatic vessels, we have these smaller lymphatic capillaries and larger collecting lymphatic vessels, but also through organ-specific adaptations that reflect the functional needs of individual tissues. So I will be focusing in particular of course, on lymphatic vessels, and in particular, these distal lymphatic capillaries that are kind of the business end of lymphatic vessels for their function, in that they are specialized for taking up fluid and immune cells from the peripheral tissues. And these vessels are very unique in their how the cells that line them are organized. And what is unique is that they have these dynamic puzzle piece like cells that you can see visualized here. And we recently found that this structural organization into these puzzle piece like shapes and their continuous dynamic remodeling is important for maintaining the integrity and kind of the resilience of this monolayer in the absence of continuous basement membrane or mural cells, or even continuous cell-to-cell junctions because they only exhibit these discontinuous button-like junctions, which are important to allow entry of fluid and immune cells into the vessel lumen. So traditionally, as I said, lymphatic vessels have been viewed as passive pathways for antigen and immune cells from tissues to lymph nodes and then systemic circulation. But now we know that they are not just passive conducts, but they also actively communicate with the tissue. And in particular, what has emerged is that they engage in this bidirectional communication with immune cells and can directly shape and regulate the function of the immune cells. So, for example, they can regulate dendritic cell maturation and migration, but they also can contribute directly to regulation of T cell functions, to regulate peripheral immune tolerance. And it has been also shown by others and also from our work that I will discuss later, that lymphatic endothelial cells also engage in this bidirectional communication with macrophage during inflammation, which is critically involved in the development of lymphatic malformations, as I will discuss. So like many others, we have also performed single-cell RNA sequencing, and this has really revealed that in addition to these structural differences and specialization of the vessels, all these different vessel types are also molecularly distinct. So here is just an example of identification in the dermal vasculature in mouse, these different types of endothelial cells from these different types of vessels. And what was interesting, we also in this study identified a unique subset or a new subset of lymphatic capillaries that we found to reside in these terminal ends of lymphatic capillaries, and what was defined by the expression of PTX3 or ventraxin-3, which is involved in immune regulation, and as well as other genes involved in innate and adaptive immune regulation, suggesting that this type of subset of lymphatic endothelial cells that we call immune interacting lymphatic endothelial cells may have a specialized role in tissue immune interface regulation. And here you can just see also in the staining for the protein that it's kind of enveloping mainly these terminal lymphatic capillaries. But so beyond this communication with immune cells and these emerging immunomodulatory functions, it is also now clear that lymphatic vessels have diverse and highly organ-specific roles in multiple tissues where they can directly interact with many different types of cells in the tissues. For example, maintaining stem cells in the hair for the hair cycling, and also in the intestine to play important roles in tissue homeostasis and regeneration. But so now I will focus on the lymphatic malformations, and want to kind of highlight what our understanding of the lymphatic vasculature has taught us that may be relevant to lymphatic diseases, and conversely what we have learned about fundamental mechanisms of lymphatic vessel regulation by studying these diseases. And so, as I said, I will focus on the diseases caused by somatic activating mutations in PIK3CA, which give rise to these solitary micro or macrocystic malformations, as well as generalized lymphatic anomaly characterized by multiple similar lesions. And so several years ago, we developed a model where we can specifically express a common causative, disease causative PIK3CA mutation, H1047R, in lymphatic endothelial cells. And we did this locally in the mouse ear skin. And this was important for us to avoid systemic effects in other organs. So this allows us much more long-term follow-up period, so we can look at the different stages of the disease as it initiates and progresses into more advanced stages. And this model, this is just how it would look like in an ear. And here in this example, on one side, in this mosaic activation, there has been only on one side of the ear a lesion developing here, where you see this highly disorganized vasculature, while here you see on the other side of the ear a normal lymphatic vasculature. And importantly, these lesions indeed progressively grow. So here we have looked at so at 3 weeks of age, we have induced the activation of the gene, and then when we follow up in the coming weeks, we can see this progressive growth of these lesions. And when we look within days, we see that this disease really starts predominantly by sprouting from these initial lymphatic capillaries, suggesting that this is really the main origin of the PIK3CA related lymphatic malformations by sprouting from the lymphatic capillaries. And so to understand better the mechanisms regulating the development of these lesions, we performed again single-cell RNA sequencing from FACS-sorted lymphatic endothelial cells. And here I just show data combined from both control and mutant mice. And here in the bottom panel, the genotypes are split, and we also see in red cells that express the mutant transcript. So we see these mutant-specific clusters enriched with cells expressing the mutant transcript in the mutant mice. And interestingly, we found that these mutant clusters expressed high levels of this PTX3, supporting the emergence of this immune-interacting endothelial subpopulation during disease progression, and potentially as an important driver of this disease. And here is just a confirmation by immunofluorescence analysis at an initial stage of lesion formation, and this advanced stage where you see this really strong expression of PTX3 and expansion of these PTX3 positive lymphatic endothelial cells. And importantly, we could also observe increased PTX3 expression in human lymphatic malformations. So in control skin, we saw it mainly sort of deposited around the lymphatic capillaries. But in the lymphatic malformation samples, we saw increased deposition all throughout, and also here quantified from several patients. And so when we then compared the PTX3 positive capillary population in the mutant and control, we found many differentially expressed genes that were related to inflammatory processes upregulated in the mutant mice, including CCL2, which was not expressed in normal lymphatic capillaries, but was upregulated in the mutant mice, which is a key regulator of macrophage recruitment. And we even saw in primary lymphatic endothelial cells where we activated PIK3CA expression, this upregulation of CCL2, suggesting that it may be even directly regulated by oncogenic oncogenic PIK3CA itself. So indeed, we then could also observe increased recruitment of macrophages in these lymphatic malformation lesions. So here shown at 2 weeks after initiation of the lesion formation and 5 weeks after, while we didn't see this for at early stages of lesion formation for other immune population. Here showed for dendritic cells a small increase at the early stage, but not at the latest stage. And these macrophages were those that produced VEGF-C, so the major pro-lymphangiogenic growth factor. And we thought that this was interesting because, of course, studies in animal models have revealed that the key signaling pathway driving both physiological and pathological lymphatic vessel growth is the VEGF-C, VEGF receptor 3 pathway, which acts upstream of the PI3 kinase. And of course, shown by many others, but also here in our own experiments, if we just in a normal mouse overexpress VEGF-C, we see this very similar phenotype, this hyper sprouting and hyper branching, and eventually this sheet-like lymphatic vasculature in the skin reminiscent of the lymphatic malformations. And interestingly, if we delete PIK3CA specifically in lymphatic endothelial cells and then stimulate these with the VEGF-C, we see that this response is completely abrogated, despite the fact that VEGF receptor 3 could still signal via its other major downstream pathway, the RAS-MAP kinase pathway. So in this context, in the hypersprouting or sprouting and growth from lymphatic capillaries, it seems that VEGF-C dependent PI3 kinase activation is both sufficient and necessary for this response. So we thought this was interesting, and we were then wondering, what if we inhibit VEGF-C in the context of the lymphatic malformations? And when we did this, we could see that this trap could quite efficiently suppress lymphatic overgrowth. And this was at the time very exciting to us and a little bit puzzling as well, since the mutation is still present here, but still to progress these lesions depend on this upstream paracrine VEGF-C stimulation. And mechanistically, we could link this, as I already showed earlier, to this pro-inflammatory signature of these mutant cells, which lead to this expression of cytokines, including the CCL2, which leads to these immune cells, and then establishes this loop between pro-lymphangiogenic macrophages and the mutant PIK3CA expressing lymphatic endothelial cells. And so thinking about this, there would be other ways of trying to now inhibit and target this paracrine signaling. And we tested a few different options. So for example, we inhibited monocyte recruitment via CSF1R blockage. So when we do this at the time when we induce the overgrowth of the lymphatic vasculature, we could inhibit the lymphatic overgrowth. It was not as efficient as the VEGF-C trap, but still we saw this reduction across all independent experiments. And similarly, just inhibition of COX-2 using celecoxib could also produce a comparable effect. So with this, we were thinking that maybe especially in more advanced lesions, this paracrine inhibition of paracrine VEGF-C could provide an added therapeutic benefit on top of targeting the PI3 kinase activity itself. And so what we wanted to then test is to allow these malformations to develop into a more advanced stage and then start the treatment when we already have a overgrowth established. And when we did this, so here are situations at the treatment end. So we see that there is a further increase in the growth of this overgrowth as been quantified here. And with rapamycin, we get actually very modest inhibition of this further growth. But when we apply VEGF-C trap, and especially the VEGF-C trap and rapamycin, we see much added therapeutic benefit. So to summarize this part, what I showed you is that PIK3CA lymphatic malformations are really characterized by predominantly the expansion of these PTX3 positive inflammatory lymphatic endothelial cells arising mainly from lymphatic capillaries. And these cells then recruit pro-lymphangiogenic macrophages that kind of reinforce this pathological vessel growth through paracrine VEGF-C signaling. And so importantly here, the disease progression is not solely driven by endothelial mutations, but also these dynamic interactions with the immune environment, which then can provide these additional targets for therapy, which may be particularly important in more advanced disease stages. So then in the second part of my talk, I would like to extend this concept to venous malformations, because I think even if we are interested in lymphatic malformations, we can also learn from other, looking at other malformation disease mechanisms, and where we find that these kind of similar principles with the mutant lymphatic endothelial cells, with the communications with their tissue environment apply. So we used a very similar model of this ear venous malformations, where we topically induce the expression of the mutant PIK3CA, but in this case we use a CRE that specifically targets the expression to blood endothelial cells. And when we look here, the venous vasculature by endomucin staining, we can see formation of these lesions, which then progressively also in this case grow. And in this case, the way how these lesions grow is very different. So there is no sprouting, but there is more this kind of bulging of these lesions as they initiate from the veins, which is kind of interesting, but we don't have an explanation to that. But it was kind of an interesting observation. And so we also we're wondering whether in a similar way, like we found the VEGF-C contributing the lesion formation in the lymphatic malformations, whether paracrine VEGF, the main angiogenic factor, could be driving the disease in these venous malformations. And we used very similar approach with the AAVs expressing, in this case, a VEGF trap that neutralizes all the soluble VEGF. But when we applied this before we induced the growth of these lesions, we didn't see any major effect. So actually we saw some effect even in the control mice, that there was reduced vessel area, but it didn't prevent the formation of these lesions, indicating that at least in these short-term experiments or short-term time frame, VEGF-C signaling is not required for the formation of these venous malformations. And so to identify similarly mechanisms driving these lesion growth, we performed again single-cell RNA sequencing, in this case, of course, from dermal blood vascular endothelial cells in mutant and control mice. And I show first just this result from the control mice, where we can perform this kind of trajectory or visualize a trajectory from artery to vein, where we see this what is called arteriovenous zonation, this gradual change in the phenotype or transcriptome across this axis. In the mutant endothelial, the mutant endothelial cells, they followed an alternative trajectory where we saw these mutant-specific clusters, which has this strongly venous-like gene signature as we would expect. But also what we found was this strong expression of markers of postcapillary veins, including different selectins and ICAM1, which are involved in leukocyte trafficking. And these mutant-specific clusters, indeed, we could confirm again that they are enriched in cells expressing the mutant transcript. Indeed, what we are looking at here, we have analyzed the expression of top 100 differentially expressed genes between venous and arterial endothelial cells, and we see that these mutant clusters shown here, the green clusters, they really have strongly a venous signature. And also by unsupervised hierarchical clustering, we can see that all these mutant clusters are most closely related to venous endothelial cells. So this is, of course, not unexpected, but what it allowed us is then to compare the gene signatures of the mutant venous clusters to the normal venous clusters in control mice. And when we did this, we found identified several genes, many of which are previously identified as targets of FOXO1, which has been previously shown to be a transcription factor which is downstream of PI3 kinase signaling, so inactivated by active PI3 kinase. And one of these genes was angiopoietin-1, which is an antagonistic ligand for a Tie2 receptor, which is upstream of PI3 kinase, and it's really a key regulator of venous identity and vascular integrity, and the key driver of venous malformations. And in normal venous endothelium, the Tie2 PI3 kinase signaling needs to be kept in balance, in check, and this is in part achieved through the opposing effects of its ligand, so the angiopoietin-1 and this antagonistic ligand angiopoietin-2. So we thought this strong downregulation of angiopoietin-2 was interesting, and we indeed could also confirm at the tissue level. So angiopoietin-2 is normally expressed in the veins, but it was completely downregulated in these lesions. We could also further show that if we drive or express a constitutive active FOXO1, it can really directly activate the angiopoietin-2. So it makes sense that if PI3 kinase is active, it inactivates FOXO1, which then removes this expression of angiopoietin-2. And why this was interesting is that we found that if we express the mutant PIK3CA in primary human endothelial cells, we could see indeed this strong downregulation of angiopoietin-2 while it's normally expressed. And this was sufficient to increase the baseline phospho-Tie2 activation, which you can see here. And if we stimulate it by the activating ligand angiopoietin-1, it could further enhance the Tie2 phosphorylation. And interestingly, if we look then in the tissue in these lesions in mice, and we have done here an assay called proximity ligation assay, where we assess the phosphorylation status of Tie2 by two antibodies, the Tie2 androgen phosphotyrosine antibody, and we could see this increased phosphorylation of Tie2 in the mouse model, and importantly also in human tissue samples. In normal veins, we saw just very little phospho Tie2, but here in the PIK3CA mutant lesional vessels, we saw increased Tie2 phosphorylation. And here is just as a positive control from Tie2 driven venous malformations. So here these are caused by mutations where Tie2 is constitutively active. So we can validate the method. So this just schematically then shown here. So we have a PIK3CA activation that kind of causes increase in the upstream Tie2 phosphorylation. So we may again have a similar situation where this upstream signaling is reinforcing the pathway activity. But we were wondering whether this reduction in angiopoietin-2 is really sufficient, or whether there is also interplay between perhaps cells producing the ligand. And we found through analysis of human protein atlas that especially in the skin, the major source of angiopoietin-1, as has been published previously, are the smooth muscle cells, which are of course surrounding these veins and venous lesions. And when we then looked in our mouse model, what we also observed that normally there are no smooth muscle cells in these capillary areas where we get these venous lesions also originating, but we saw this strong ectopic recruitment of smooth muscle cells around these abnormal lesions, which is just quantified here. And interestingly, we found that angiopoietin-1, which you can see here, which is normally expressed in venous, in a subset of venous smooth muscle cells, it was strongly expressed in these developing lesions, which are here also proliferating, so stained here with the EDU to mark proliferating cells, both in larger veins and also in these smaller venous capillaries. So you can see this angiopoietin expression, suggesting that we have a situation where indeed we shut down the antagonistic angiopoietin-2 ligand while we have an increased expression of the agonistic ligand through these smooth muscle cells. We could also show that in human samples, there is a heterogeneous smooth muscle cell coverage with some regions which are indicated here in red lacking smooth muscle cells, while others showing this excessive multi-layered coverage, which suggests that smooth muscle cells as a potential source of angiopoietin-1 may accumulate also in these lesions and contribute to increased Tie2 signaling. So again, this was interesting because it suggests that we might have another way of inhibiting venous malformations by limiting this angiopoietin-1 Tie2 signaling, which may offer more specific means compared to inhibition of PI3 kinase. And so when we then tested this, we first again wanted to test this in a context of more advanced lesion where we normally with rapamycin observe only a very limited therapeutic effect. But when we now inhibit or use a Tie2 inhibitor, we see much better effect, and especially when we combine this with rapamycin, we see almost a complete blockage of the growth of these lesions. And if I have a few more minutes left, I will just show a few more slides where we did an analysis of clonal growth of these lesions using a clonal multicolor lineage tracing system that enables mosaic labeling of cells. And with this approach, we can really reveal that these lesions, when they initiate either in larger veins or these venous capillaries, they primarily originate through this clonal expansion. And the key advantage of using the ear skin is then the ability to combine this with longitudinal intravital imaging in live mice. And so we have here tracked the lesion evolution over a 2-week period where we can visualize directly the clonal expansion within the developing malformations. And here are just tracking some of these clones. And so what we can now then do with this platform, that we can also assess in real time therapeutic responses. So we can first allow, as shown here in the pre-treatment, we can allow lesions to grow, and then we can start a treatment, and then at the end of the treatment, we can come to exactly the same point. And then we can calculate the number of cells in this case, which is then plotted here. And we can see that while in the untreated mice, they continue to expand, those that received the combination therapy with rapamycin and Tie2 inhibitor, they showed this growth arrest, or even in some cases reduction of cellular content. So as a final note, I will just come back to the lymphatics, but we can also see that the similar principle applies in at least in venous malformations, in PIK3CA-driven venous malformations, where we saw that these endothelial autonomous mutations really initiate the disease, but they are not sufficient to explain progression or clinical severity of this disease. So instead, the disease evolution is critically then modulated by these paracrine and even systemic interactions with the tissue environment. So in case of the lymphatic malformations, in particular with the immune compartment. And so this makes the microenvironment not just a kind of a modifier, but also an important potential therapeutic target. And so if we can better understand these mechanisms and these multicellular interactions, we can hopefully develop better treatments. So finally, I would just like to thank the people who have done the work. So most of the work has been done by people who have already left the lab. Especially on the venous malformations, Marle Kraft, and the lymphatic malformations, Milena Petkova and Ines Martinez Coral, and now continued by Vishal Mohana Krishnan. And of course, we are very grateful to all our collaborators, especially for all the clinical samples and also mouse models and AAV vectors and other important tools from many colleagues. And of course, funding. Thank you for your attention also. So, and I'm happy to take questions.
Thank you, Taija, for a great presentation. So the floor is now open for questions, and I'll get the discussion started. So I actually have 2 questions. So the first is, I think it's amazing how different endothelial cell populations are sensitive to mutant PIK3CA, whereas some seem to be more sensitive than others. And I was curious as to your thoughts as to why capillary LECs seem to be more sensitive to mutant PIK3CA than like collectors.
Yes, I don't have the answer, but we have been also wondering about that, and um, we were wondering maybe whether naturally, there is a difference in the sort of baseline levels of RAS versus RAS MAP kinase versus PI3 kinase activity, and then maybe if this population is not normally sort of known to handle that sort of pathway, when it suddenly comes, they just sort of overreact to it, while maybe some other cell type is more able to handle it. Of course, it also may be like, if you think about it in the context of lymphatic vessels, the collecting vessels and capillaries are very different. So the collecting vessels have more basement membrane and smooth muscle cells. And what we see that in the lymphatics, the main really immediate response to PIK3CA is this sprouting and migratory response. So it could be also that the collecting vessels are more constrained due to this sort of basement membrane and so on. But yeah, we don't have the answer, so it's just speculation.
And I'm going to sneak in one more question, even though we have a few hands up. And that is, so the underlying mechanisms of the PIK3CA mutations vary depending on whether or not it's a kinase domain mutation or like a helical domain. And I'm curious if you've looked at helical domain mutations and the dependence of those on upstream receptor tyrosine signaling.
Yeah, I think it's interesting because indeed, in the context of cancer, it has been shown that this requirement for upstream is different. And so we haven't looked at that, but it would be definitely something interesting to
right.
see.
And we'll move on to Dr. Ostuara.
Yeah. Hi, very, very nice talk. So I'm just going to ask a really kind of an obvious question about the non-genetic kind of loops, the part of it. So part of the drug response that we see, like with rapamycin and everything, like the effect of it probably can be explained by this like non-genetic loops that you just described. I was just wondering, can we have some sort of an intervention in that non-genetic part of it so that the entire thing is now dependent on the mutation itself, so that we can kind of create a synthetic dependence back to the kind of PIK3C pathway by intervening the non-genetic part?
So it may be difficult when the disease is really kind of at an advanced stage, because there are so many changes in the tissue. So for example, what we see with the reporter, where we can really precisely identify the mutant cells and normal cells, we can see that at these advanced stages also the wild type cells, I'm talking now about the lymphatic malformations, the wild type cells also start to proliferate. So there are many complex interactions. So I think that for lymphatic malformations, based on, of course, just on our mouse model, really the inflammatory sort of response is really critical. And whether you can completely sort of abolish that and in that way return the tissue to a normal state, I don't know if it's really possible. But I think like this sort of immune, something that, immune suppressive treatment on top of targeting the PI3 kinase would anyway give some kind of benefit.
Thank you.
All right, we'll go Celine and then Shoshana.
Great talk, Taija, as always. I'm going to go back to the signaling part, following up probably Mike's. Given the FOXO1's role in both the PIK3CA and TIE2, feed-forward circuit. I was wondering if the lymphatic valve identity might be the dysfunction, what you're seeing, instead of an early function instead of downstream consequence of the gross structural changes. Do you have any idea about that?
So in the PIK3CA mutant mice, we don't see any effect in the valves. So yeah, it's quite peculiar.
Okay. Yep, great,
yeah.
thanks.
Okay, thank you for a great talk. My question is, clinically, what you see in the patient is that sirolimus, in response to sirolimus, many lymphatic malformation regress, irreversibly regress, whereas venous malformations do not. They are less symptomatic, but I never saw regression. Do you have any explanation from your experiments?
So I guess it could be that the sirolimus effect, so it affects basically both the downstream of the PI3 kinase, but it will also affect the immune cells. So I think it would be interesting if it would be possible in the patients to better characterize the sort of immune response. How much of the effect could be also partly because of the effect on the immune system. So it kind of already targets the both of them.
Okay, thank you.
Well, if there are no additional questions, we'll thank Taija once again, and we look forward to seeing everyone at our next presentation. Thank you.
Thanks so much.
Mhm.
Bye-bye.